Somewhere in the ocean lives a creature that never has to die of old age. It simply hits rewind. They call it the immortal jellyfish, but most never survive long enough to use the trick. So what exactly is it resetting, and why can't it save itself? In 1983, German embryologist August Weismann dipped a net into the sunlit waters of the Bay of Naples. He pulled up a translucent bell-shaped hydrozoan measuring just 4.5 millimeters wide, barely the size of a pinky nail. Weismann documented the tiny creature under the scientific name dohrnii. To him, and to generations of researchers who followed, this miniature predator was just another obscure marine specimen. For over a century, its mobile adult form, the free-swimming Medusa, floated through the Mediterranean entirely unnoticed by science. Its famous nickname, the immortal jellyfish, did not yet exist because no one had any reason to suspect its bizarre secret. What happens when a living creature does not accept the usual ending? In the late 1980s, German marine biology students Christian Sommer and Giorgio Bavistrello kept these tiny jellyfish in laboratory tanks. They expected the adult Medusae to spawn, age, and die. Instead, when the animals faced physical injury, starvation, or sudden temperature and salinity changes, they witnessed an eerie biological contradiction. The jellyfish refused to die. Within 24 to 36 hours, the adult bell contracted, absorbing its own tentacles and dissolving its mature physical structures. It collapsed into a tiny, unmoving cyst-like blob on the bottom of the tank. From this apparent decay, the mass sprouted root-like stolons, anchoring to the surface and rebuilding a brand new polyp colony. It was a complete chronological U-turn, officially recognized in the early 1990s. The butterfly had crawled back into its cocoon to become a caterpillar. What does rewind mean inside a living body? How does a specialized cell forget what it is? Inside that contracted cyst-like blob, the jellyfish undergoes transdifferentiation, a rare process where fully mature cells bypass standard biological limits, converting directly from one specialized type into another. Unlike ordinary healing, which merely patches up a wound with matching tissue, this is a wholesale identity shift. Striated muscle cells and nerve endings dismantle their own structures, losing their adult Medusa identities to become the completely different cells required to build a polyp. It is not time travel, but rather a profound biological recycling program, akin to a butterfly dissolving its wings to reconstitute the body of a caterpillar. This cyst is no dormant pause. It is a hive of active remodeling. Scientists debate whether this occurs through direct cellular conversion or by activating a pool of flexible interstitial stem cells that reprogram the tissue. Regardless of the exact pathway, the resulting polyp colony eventually initiates asexual budding. Within 2 to 3 weeks, new buds form on the branching stolons, and in 4 to 6 weeks, they release a generation of young, genetically identical medusae into the water column. Under controlled laboratory conditions, this loop has proven remarkably durable. Biologist Shin Kubota maintained a single colony for 2 years, observing 11 complete rejuvenation cycles. Other cultures have reached 10 to 14 successive resets without any drop in regenerative capability. Theoretically, as long as water quality, temperature, food supplies, and cyst formation remain ideal, this loop can repeat forever. Yet this genetic carbon copying does not erase previous physical scarring or copy-acquired injuries. It is a clean slate, a theoretical blueprint for eternity. But a blueprint in a sterile lab is very different from survival in the open ocean, where the reset button must face the brutal realities of the wild. In the open ocean, biological immortality is not invincibility. Scientifically, it is defined only as the absence of senescence, the technical absence of an unavoidable increase in death risk from aging. Immortality, it turns out, still has a predator problem. Fish and larger cyanidarians routinely consume these fragile floating bells long before they ever have the chance to reset. Plankton surveys estimate that up to 70% of juveniles are lost to opportunistic predation, and no wild individual has ever been documented surviving for centuries. Beyond hunters, physical limits are absolute. While acute deprivation triggers a survival response, prolonged starvation remains quietly lethal. Disease, severe injury, extreme temperature spikes, or a failed cyst formation will always kill the organism outright. If the trick is real, what protects the cells that perform it? In 2022, a comparative genomics team led by Maria Miglietta published a study in the Proceedings of the National Academy of Sciences. Titled Comparative Genomics of Mortal and Immortal Cnidarians, the research compared the immortal jellyfish directly with its mortal cousin, Turritopsis rubra. The study revealed that Turritopsis dohrnii possesses a genome of approximately 390 megabases containing 17,468 protein-coding genes. By contrast, the mortal relative Turritopsis rubra has a smaller reported genome and a significantly lower gene count. Within this genetic blueprint, researchers identified extensive gene family expansions, a term defining the presence of additional related copies or variants of specific genes. These expanded families are linked to DNA replication, DNA repair, antioxidant stress responses, and stem cell flexibility. Most notably, the expansions safeguard telomeres, which are the protective structures at chromosome ends that normally wear down as cells divide. The jellyfish carries a specific variant of a gene called POT1, which displays reduced telomere binding, suggesting a pathway that keeps these chromosomal caps intact during the reversal process. These genomic discoveries are associations rather than outright proof. Sampling remains limited to a small pool of individuals, and functional validation of these genes is incomplete. This molecular toolkit does not mean humans are close to escaping biological aging, nor does any direct human longevity therapy follow. Instead, the potential applications lie in stem cell medicine and cell regeneration, focusing on cellular reprogramming and functional tissue restoration rather than copying the jellyfish life cycle into humans. To move beyond theory, researchers must conduct functional gene tests, long-term telomere measurements, broader population sampling, controlled cell fate studies, and rigorous checks to ensure rapid regeneration does not lead to uncontrolled cell growth. Nature has built a flawless reset mechanism, but science is only beginning to read the manual. Subscribe to explore more stories of creatures that defy nature. The jellyfish unlocked the biological secret to eternity, yet remains utterly fragile. Nature's ultimate irony is writing the code for immortality in a creature easily swallowed whole.